Good morning. I will give the talk in English because of both languages today, and as you have seen yesterday, the switch between 3 languages was a bit painful for a French brain, so I will give it in English. So physiology of pigmentation, but mainly what is melanogenesis and what is UV-induced pigmentation, because I think this is crucial for a dermatologist to know the aspect of the molecular basis to induce pigmentation. So at the first level, you have the melanocyte. Melanocyte is originated from the neural crest, and is located at the basal layer of the keratinocytes and associated with around 30 keratinocytes through dendrites, and through these dendrites you have the transmission of melanosome. Melanosome is the specific organelle of pigmentation. So the organelle is transported through the dendrites with a very sophisticated mechanism. I have to move the, it's okay? Okay, sorry. I thought that it was wrong. Okay, so it's a very sophisticated, it's impressive to see how sophisticated it is to transmit these organelles to the keratinocyte. And this is also induced by UVR and mainly UVA also. It's interesting to see that, in fact, when you have been to the sun, you have an immediate pigmentation, and this immediate pigmentation is given by a immediate transmission of the melanosomes through the keratinocytes. So this is related to this system. So the melanosome is transferred, and it becomes, the originated story of melanogenesis is a key enzyme, tyrosinase, that is also the synthesis happen inside the melanocyte. You have also the synthesis of the melanosome matrix, the transport of tyrosinase and the formation of the melanosome, and progressively the transfer of melanosome into the keratinocytes, and then the melanin is removed by desquamation. So this is what happens, in fact, when you go on holiday. You are exposed to sun and you get a tan, but after that the tan progressively disappear because of the melanin that is disappear with desquamation. So a key enzyme, tyrosinase transforms tyrosine into dopaquinone, and then you have the association or a sulfhydryl compounds, and this gives pheomelanin. This is the red melanin, or you see that it goes into a specific compound which is dopachrome, and this gives the synthesis of eumelanin. So 2 major forms of melanin that are produced into the melanocytes: brown-black eumelanin and yellow-red pheomelanin. So I give the image of my mice. I like them very much. So 2 major forms of melanin, and you can find that again into mice, and they are models to study pigmentation. And the skin pigmentation correlates directly with the eumelanin content, which is more abundant in dark and highly pigmented skin. And now I would like to explain to you the difference in the skin. But first, the history of the melanosome, that is progressively, you have the formation of the melanosome, inside which you have tyrosinase who synthesize melanin, and also what is interesting to know that you have specific melanosome, eumelanosome and pheomelanosome. They are not the same size, the same shape, and if you look with electronic microscopy, you can really differentiate them through the keratinocyte. So what are the phototype? You see that this table, I'm sorry, it's in, I go into this, it's in French. So you have the melanocytes, the keratinocytes, and the keratinocytes at the basal layer and at the superficial layer, and to see the difference in the phototype. So phototype 1 and 2, you have melanosome with pheomelanin. Within the keratinocyte from the basal layer, you have few melanosome, and then you have nearly no melanosome. And when you go through the phototype, you see that, in fact, the melanosome persists for the dark skin until the corneal layer and gives a pigmentation to the skin. So, in fact, if you compare both skin with a fair skin and a dark skin, that you really have the difference into the number and the type of melanosome. So this is also a summary. Main of this excellent summary are coming from the Bologna. Jane Bologna did her PhD in pigmentation, and so she did really a very nice chapter on pigmentation. So you see the difference between the dark and the fair skin. So the major determinants is the quantity and the quality of the pigment production, but not the density of the melanocytes into the different type of skin. So you know as much the incidence of skin cancer correlates inversely with skin pigmentation, with the highest incidence with the people with light skin color. And the best evidence was given by the Celtic population, all the English people, the people from UK who migrated to Australia, and where the incidence totally changed about the skin cancer, because they were exposed to the most aggressive UV in Australia. And so this is one of the proof that we had that skin cancer was induced by UV exposure in light skin. So a very important element to know about pigmentation is one receptor, MC1R. MC1R is a MSH receptor, and these receptors can be stimulated by MSH, alpha MSH, and this makes that the stimulation give the production of favor the production of eumelanin instead of pheomelanin. You see also something that is amazing is agouti. We'll see this a bit later on. You all know the difference in color with Labrador, for example. A Labrador with a very light fur is with agouti, and if it's not, it's a black fur. So this makes the difference also for the dogs. So it's interesting to know. But in the case of human, it's the allele variance that makes that the function of these receptors change, and makes that some of our patients has got, or humans, has got red hair and a fair skin. So, but I wanted to show you this because this made me a shock one day. The animals, like a zebra or a white bear, has got a black skin. This is amazing because I realized, and I was quite old when I realized that I never thought about the color of the skin of the zebra and also about the white bear. And in fact, we were also black. We are all originated in Africa, and you know the story about Lucy and our common ancestor. And so it's interesting to know for all the story about ethnic skin that finally we were all black. We were all black. We migrated through the world to Asia, to Europe, to Australia, and then we lost our pigmentation. And this is an adaptive process. to regulate, in fact, the UV penetration into the skin. And why? Because we need vitamin D. So we needed to receive the UVB to transform the vitamin D and to be able to live, and you know how much vitamin D is important for our life. So the process of the loss of pigmentation is really related to an evolutionary gain for humans. So, but one thing I wanted also to remind to you today is how does UV induce pigmentation? And when I began my PhD, in fact, it was not known. And it's so strange because it's so normal to get tanned by the sun. And it's quite recent that that was discovered. So first, we know that UV exposure induces an acute process with inflammation. It can be the sunburn, immunosuppression. We know how much we use this also as a therapy. The epidermal thickening, we know that acne is increased after UV exposure because also of the epidermal thickening. And finally, the tanning. And the chronic exposure, finally, exposed to results in photocarcinogenesis and photoaging. So it's interesting to review the intrinsic protective mechanism and what is induced by UV. So by the sun exposure, you have something that doesn't change, is the hair fur. So it doesn't change, we don't change the color of our hair, but we change the pigmentation of our skin. We increase the thickness of the stratum corneum, and we induce also antioxidative enzyme. We induce also DNA damage, so we need to have a DNA repair, and this is also activated by UV. We need also the cell cycle arrest, just to be able finally to repair, and finally it induces mutation and the removal of mutated cells. So when we are exposed to UV, it's a generation of oxidative stress that induces also DNA damage and lipid peroxidation, but also the induction of photoproducts. And these photoproducts are very important. Why? Because it will play a role, and that I will explain a bit later. But first, I just want to remind you the MC1R, so the MSH receptor. This is also stimulated by UV on itself. MSH, as I just mentioned. Also, you know that inflammation can increase pigmentation of the skin, and you see that IL-1 is induced also by UV and related to the MC1R, and it's inhibited by TNF-alpha and TGF-beta. But we won't go into this mechanism, which are much more complicated and not needed for today. But we will go back now to this general molecular pathway of tanning that resume all the aspects of tanning. So, in fact, and I will go back to all these mechanisms. So, UV-induced DNA damage, and you see that you have, that was just mentioned, different elements and growth factors that can be induced by UV, including also MSH. We will review also all these mechanisms induced by the DNA damage that will induce, in fact, pigmentation. And there we have one element that's very important for the melanocyte, MTF pathway. And finally, we will increase the production of pigmentation inside the melanosome and the transfer of that melanosome into the keratinocytes, as I just mentioned before, within which the melanosome we have the pigment synthesis. So UV damage induced P53. You see that this publication is in 1997. So it's quite recent that we understood, and it was given by Barbara Gilchrist, who showed that, in fact, P53 was crucial induction of by pyrimidine dimers. And finally, P53 promotes the transcriptional activation of POMC. Alors, POMC is propomelanocortin, and we'll see that then why we have such a pigmentation. But so it's a crucial aspect of pigmentation. POMC is a direct translational target of P53, and it's directly controlled by P53. So what's POMC? It's a precursor that is cleaved, and I will ask you, what do you find in this molecular image that reminds you something? What do you see there? Which molecule do you see that can ring a bell? So, this level, what do you see? Maybe you don't see it. So, ACTH. What's the disease where you get pigmentation? Addison disease, okay? Then you see this. Alors, I understand that it's not so easy. ED, endorphine. You know, when you go to the sun, you have this feeling, good feeling, why people can get finally addicted to tan. This is the reason. So some people are really related to this mechanism. And finally, MSH. Okay, so it's important to understand that you have basics that can explain to you some elements of pigmentation, but also pigmentation related to disease. So MSH, ACTH, and endorphin, three elements that are very important with the POMC. So this first summary gives a bit what I just explained to you and summarize a bit all the elements of inducing pigmentation. And finally, we have UV induced by pyrimidine dimers. Pyrimidine dimers induce P53. P53 will activate the transcription of POMC, and because it contains a consensus sequence. And finally, MSH, and this is summarized here. MSH will stimulate pigmentation through the MC1R receptors and induce the formation of melanosome, and melasome will be transmitted to the keratinocytes. So a whole story of your pigmentation when you are during your holidays. Okay, thank you for your attention.
Thank you. Very inspiring and super interesting. Is there any question for Professor Del Marmol?